Welcome to the unit on genetic variation. In this section, we will discuss what DNA mutations are and how they occur, various types of mutations at both the DNA and protein level, and what effect those mutations can have on the genotype and phenotype of an organism. Although it is an important topic, we will not be discussing the details of DNA repair mechanisms. It is important for you to realize that not all DNA damage will become a mutation, because the cell is able to repair many DNA mistakes. However, you will not be tested on the details of those repair mechanisms in this course. This video will focus on what mutations are and how they occur in DNA. A mutation is defined as any heritable change in the sequence of the genetic material. Heritable means that the change in the sequence is stable and passed on through cell division. It is mutations that account for the genetic diversity of populations and the genetic uniqueness of every individual. Some of these mutations occur randomly in the cell during DNA replication because DNA polymerase is not perfect and in eukaryotes makes a mistake at a rate of about 1 nucleotide in every 10 billion nucleotides that it replicates. You'll notice on the graph that viruses mutate at a much higher rate, especially if their genome is RNA, because RNA is a much less stable molecule than DNA. Additionally, there are exceptions to the 10 to the minus 10 rate, such as certain areas of the chromosome called hotspots, because they pick up mutations more often. In multicellular organisms, there are two different cell classifications, and mutations can occur in the DNA of both cell types. Somatic cells are all the cells of the body that are not sex cells, such as your skin, liver, or heart cells. If a mutation occurs in a somatic cell, it is passed to the daughter cells of that cell during mitosis, but is not transferred to future generations because it is not in the egg or the sperm. The egg and the sperm are gametes, also called germ cells. If a mutation occurs in these cells, then they will be passed to the next generation. Oftentimes, there are many rounds of DNA replication that have occurred before a germ cell is formed in meiosis. For example, in sperm, the cells of the testes must first go through many rounds of division to form the organ, and then more rounds in the production of sperm. Each round of replication is a chance to pick up more mutations. So in a human male, by the age of 30, about 400 cycles of DNA replication and cell division have occurred. This produces a mutation rate in sperm that is about 400 times greater than the overall rate of mutation per single cycle of replication that we looked at in the previous chart. Instead of graphing the mutation rate per round of division, like the first chart did, you can also graph the mutation rate per generation, or the number of mutations that can be expected from one generation to the next. This graph takes into account the number of germline cell divisions as well as the genome size. Genome size is important because the larger the genome, the more opportunity there is for mutations to occur. When we look at the mutation rate this way, we can see that the mutation rate for humans per generation is approximately 30 mutations per gamete. This means that there are an average 60 nucleotide differences between you and each of your parents. Luckily, most of these mutations are considered neutral. They have no effect on the cells because only 2.5% of the human genome actually encodes for protein. If a mutation occurs in the non-coding DNA, it often will not affect the proteins and their functions. This mutation rate per generation is most important when looking at germline mutations, while the original rate of mutation during replication cycle is most important when looking at somatic mutations. Somatic mutations will affect the traits of the individual, but they will not affect the traits of the individual's offspring. We have already discussed one effect of somatic mutations when we discussed mutations that can affect cell cycle regulation. The majority of cancer-causing mutations are somatic mutations, leading to the development of abnormal cells in the individual, but not in the germline, as seen here. Another example of somatic mutations can be seen in this Japanese morning glory flower. A pigment-producing cell has picked up a mutation, and now all of its daughter cells produce a purple color instead of white, leading to a small portion of the flower looking different from the rest. It is important to understand that mutations arise spontaneously. They are not occurring to allow an organism to adapt to a changing environment. This was shown in an experiment done by Joshua and Esther Lederberg. using bacteria and antibiotic resistance. They first grew bacteria on an agar plate 1, which had no antibiotics, so all the bacteria were able to grow. When bacteria grow on a Petri plate, they form spots of growth called colonies. The scientists then used a process called replica plating, where they take a velvet cloth and press down on the growing colonies. They are then able to transfer those colonies to a new Petri plate by pressing the cloth down onto another Petri dish. However, this second agar plate is different than the first one because it contains an antibiotic. This antibiotic should kill the cells. After incubating the plate, the Lederbergs found was that not all the bacteria died. At least one colony was able to grow on the plate. Because of the replica plating technique, they are able to go back to the original plate and find the original colony growing in that location. This is important because they wanted to know if the mutation that was allowing the bacteria to grow in the presence of the antibiotic was already present in the bacteria before it was exposed to the antibiotics, which means it was just a random mutation, or did the mutation occur after the cells were put on plate 2 in response to the environmental change of the antibiotic. To test this, they took some of the original colony and grew lots of cells which would be genetically identical to each other in media without antibiotics. They then transferred the new culture to a plate with antibiotics. If the mutation was present in the original cell, then the cells in the broth would have that exact same mutation and lots of cells should grow on the plate. This is exactly what they saw, indicating that mutations occur randomly, not in response to the environment. The single colony on the original plate just happened to have a random mutation that turned out to be beneficial and allowed it to grow when the environment changed. Spontaneous mutations that occur because of errors by DNA polymerase are not the only way that mutations can occur. The mutation rate of the DNA can be increased, which means the mutations happen more often if the cells are exposed to a mutagen. A mutagen is something that causes damage to DNA, because that damage often leads to the formation of a mutation. Things that we consider carcinogens, or cancer-causing substances, are all mutagens. They can cause cancer because they increase the mutation rate of DNA, which increases the chance of a mutation happening in a tumor suppressor or proto-oncogene of the cell cycle. Some examples of mutagens are X-rays, which can cause breaks in the sugar phosphate backbone in one or both strands, UV light, which causes thymine bases to cross-link together, preventing their base pairing with the opposite strand, bleach or hydrogen peroxide, which causes bases to be lost from DNA, and various chemicals such as those found in cigarette smoke, which can add bulky side groups to the bases, resulting in improper base pairing.